Oh, sorry. Just made your face super huge. Hold on. There we go. Let's go back to this thing. There we go. That looks right. All right, so I think we're streaming on YouTube and we are recording. So I want to welcome everyone to our next Water, Wetlands, and Watersheds seminar. So just as a reminder, we're celebrating the 50th anniversary or the 50th birthday of the Center for Wetlands here at UF. It was started in 1973 by Howard Odom. I never had a chance to meet him, but he was a father of the ideas of ecological engineering and systems ecology. And through his work and his students' work, even now his students are beginning to become retired. Wetland ecology, systems ecology, and ecological engineering were advanced. So we have some alumni coming back to speak with us this semester. So I'm going to introduce Dr. Kelly Chinners-Reese. She's a wetland community ecologist interested in landscape connectivity. She has her bachelor's in forest resources and conservation and both her master's and PhD in systems ecology here from UF. Kelly is a professor of environmental science, policy, and management in the School of Science, Technology, Engineering, and Math at American Public University. Kelly serves as faculty co-advisor for the National Association of Environmental Professionals, the Wildlife Society student chapters, and she co-leads the Water Testing and Awareness Project. She's a certified senior ecologist through the Ecological Society of America, and she'll tell us about a lot about it today, but she has managed the Florida National Wetlands Condition Assessment all the way back to when it started in 2011. Kelly's research interests include indicators of wetland condition, and past projects have included geographically isolated wetlands, restored and enhanced wetlands and springs, wetland mitigation banking, and characterizing reference wetlands and wetland damage and deterioration and how we can get them back. So with that, I will turn the floor over to you, Kelly, and make sure we are good on the YouTube. We are good. All right, go for it.
All right, great. Well, thanks so much, David, for the introduction and to everyone for inviting me in today. It's exciting to have the 50th anniversary. As Dr. Kaplan mentioned, I had a little bit of time overlap with H.T. Odom, about 4 years of my studies there. So it's really exciting to be here and to be able to present some of the work that we've done. Today, I wanted to talk about the National Wetland Condition Assessment. And this project is really enormous. It is nationwide and many years since it's been going on. So I feel like it gives us a really nice opportunity to dive into some very interesting topics in wetlands and to kind of go from there. I would be crazy to start without thanking and acknowledging just the depth of this project. So as David had mentioned, we've been doing this project in 2011, 2016, and 2021. These were the very large field efforts that happened. There's a lot of behind the scenes that go into that to try and get the field crew where they need to be. And then, of course, on the other end of that, there's a ton of work on processing all of the samples and working through the data as well. So the list of folks here are those that have participated specifically in the field portions and planning for UF. And this is just one state in a nationwide survey. So you can imagine really the depth of what goes into this kind of project. I wanted to do those introductions right away, just so you understand like how much goes into this. All right. And so as part of sort of that whole mindset of being in the 50th anniversary of the Center for Wetlands, one of the images that has always stayed with me is this idea of the macroscope. Some of you may be very familiar with this. It dates back to a book published first in the 1970s, and it's been republished since then. But the idea here is that when we look at the complexity of natural systems, we use a macroscope because it allows us to really filter out all of the complexity, take those details out and really eliminate them, put our framework and our reference into this simplified model, and then from there, try and understand what is happening in the system, the functions, the feedbacks, the connectivity and processes. And so it's within that framework that I've been working in biological indicators work and wetland condition. And in my opinion, the NWCA project is just a fantastic playground for these kind of ideas and to try and understand all of the functions that are around us, in particular for me in wetland environments. The sort of graphic that I have down here on the bottom right, this is my trying to take a more complex world and ordinate it, right? So you can think of these different gradients or axes to talk about conditional changes, environmental variables, and within it, we can measure different components of a system. And so what we're trying to do is sort of mash those together, right? To be able to gather data from the field and understand and make sense of the complexity of natural systems. So to give you a big overview kind of picture, the National Wetland Condition Assessment is just a small part of a much larger project called the National Aquatic Resource Surveys, or NARS. It's an EPA project, and it combines EPA, of course, other agencies like NRCS with states, tribes, and on here there should also be like national history programs, universities, Great Lakes Environmental, all kinds of contributors to these large projects. And on the map here, this is just really the first survey runs for a National Lakes Assessment, National Rivers and Streams Assessment, National Coastal Condition Assessment, and then down there, National Wetlands Assessment 2011. And this survey has now been repeated in its entirety at 3 iterations. So not only do they have all of the field sites you're seeing up there, but they have 3 times as many. So this is just really an enormous pool of data on our aquatic systems that we have available to us. I've left the link here on the webpage. Anybody can go and grab most of those data available and start really diving into this. And that's one of the real benefits I think of NARS, is that all of these data are publicly available to answer all kinds of questions on large scale aquatic system studies. So specifically, the work that we've been doing through the University of Florida Center for Wetlands has been on the Florida portion of the National Wetland Condition Assessment. As I mentioned, it's mostly the lower 48 states. There are some studies that have been done outside of those boundaries as well. And it's really a collaborative survey. So we're looking at physical, chemical, and biological integrity, which meshes very well with the wording of the Clean Water Act, for those of you familiar with it. We're also thinking about the foundation of the NWCA as coming from something called EMAP, or the Environmental Monitoring and Awareness Assessment Project. And that was in the late 90s, mid 90s through the early 2000s. And that really set the foundation for how can we scale up a nationwide assessment of wetland condition. And if you'd like to, you can go and see the EPA's overview. I'm not going to give you an overview of all of their findings from the last surveys, but the EPA has that available for the first 2 wetland surveys and all of those other surveys if you're interested in rivers and streams and lakes and coastal wetlands, coastal areas. All right, so within the NWCA specifically, I needed to kind of give you a framework for how I'll be talking about some of the wetland data that we're looking at. Now, the EPA has taken the more characteristic Howard classification, some of you are familiar with, and has really simplified it a bit. So they have just estuarine herbaceous and woody, or inland herbaceous and woody. And that pools all of the different, in a lot of cases for the inland systems, hydrologic regimes together to characterize the vegetation communities. They do so across 4 different regions. Now, a lot of my interest has been coastal plain related. I have a lot of background in Florida and coastal systems. But I do want to let you know that there are data all across the US, and that's also something that I know AJ is using in some of her work that I'll mention just a little bit as well. The map at the top is showing you only the 2011 and 2016 survey points. I didn't have the 2021 to put on there as well, but it gives you an idea of all of the areas that are being covered by this survey. There's a little bit in some of the mountainous regions like the Appalachians that don't have wetland samples. There's some in the desert Southwest, but for the most part, you can see a pretty extensive coverage of data across the lower 48 states. And just to give you a picture of the size of this survey, between 900 and 1,000 survey sites are done in each of the survey years. There's also about a 10% resample in some of those years, at least for Florida, it's about a 10% resample. And there's the ability then to look at data from 2011 and the same exact wetland, 5 years later, and sometimes 2016 to 2021 as well. Some of the data is outstanding still for 2021, going through quality assurance, quality control checks. So not all of that is available to us right now to use. But we still have a huge amount of data that we're able to wade through. One thing that is important to note, I think, here for some of the analyses is that on average, somewhere between 50 and 60% of the sites of the wetlands on any given day when we go out to sample actually have standing surface water. I think that's sometimes a misconception about wetlands because they do go dry and we can't always get suitable water for sampling. And so just something to keep in mind, our sample size goes down dramatically when we're talking just about water parameters compared to some of the soils or vegetation. All right, so those of you in Florida should be familiar with this photo here, but this is all of the sites that have been surveyed across 2011, 2016, and 2021. We have 176 unique sampling events and 154 unique sites. And that means 22 of our sites we have revisited at some point. So say you were sampled in 2011, we also sampled you in 2016. There's about 12 of those. And then also from 2016 to 2021, another dozen or so have been resampled. You can see we went all the way to the border of Alabama, all the way to the border of Georgia, down to the Florida Keys, all across the state. So there's some in Everglades National Park and Big Cypress. There's some in urban areas like in Tampa. Basically, there's just a lot of information here. Looking at this map, you might think about recent events like Hurricane Idalia. There's a lot of data here that could be used in some kind of future study to think about what are we seeing, how are things changing in the landscape. So there's a lot of big questions we can ask with NWCA data that go beyond the original scope of monitoring for the project. I don't want to spend a lot of time on methods, but I did want to let you have at least an example of what it looks like on the ground. If you were going to go into one of the assessment areas, the circle on your right shows a 1/2 hectare circular assessment area, or AA, and I call this the site scale. And this is where we do vegetation sampling. Those are the gray quadrats. We would take a water sample. It doesn't have to be located where I've put it there on the diagram. It has to be in the most suitable spot within that 1/2 hectare. And we have a soil pit as well. And then the nice thing about NWCA is it also takes a step back and it moves from beyond that site scale and it looks at a landscape effort. And that is through buffer plot analyses. And on each of the cardinal directions, you go out and you make observations on disturbances, invasive vegetation that might be present, and that kind of thing as well. So not to spend too much time on methods, those are all very well documented in EPA information. But just so you have an idea that when I'm drawing things in circles, it's because in my mind, that assessment area is this nice little 1/2 hectare circular plot. And there's a lot of information that are gathered, and it's a one-day sampling event. And there's information on vegetation, hydrology, water chem, algae, buffers, and then also some more detailed soil analysis. All right. And then what EPA has done with this is they've gone through and they've decided on different types of indicators to look at. And again, these tie directly back into that Clean Water Act concept of physical, chemical, and biological integrity, really trying to connect what we're measuring on the ground with what it means to manage these systems and what policy decisions we might be making. So outside the scope, perhaps, of the NWCA, which is a monitoring program, but the implications for these data are very wide-reaching. So how does the NWCA fit into this framework? So again, as part of this 50th anniversary celebration, I go back to this idea of wetlands as a systems. What is the systems view of the wetland? And one thing that has really been formulated for me in systems thinking is that not only do we need to look at the scale of interest, and so in this case, I would say it's that assessment area or that site scale, but we need to take a step back and really look at what's beyond that assessment area. What is our landscape scale? And then we can zoom in as well, and we can look down at that local scale. What's happening in the nuances of soil chemistry and heavy metals, and what's going on with the water chemistry. And so those are the sort of connections and concepts that I think are very well suited to this data set. Part of that framework comes from this idea that wetlands themselves are nonlinear systems. We think of them as being self-organizing. They're certainly experiencing pulse dynamics, from flooding to droughts to invasion of species to grazing events. There's all kinds of things that are causing, unequilibrium in these systems, really, and those make for very interesting case studies as well. Sort of thinking about adaptive nature, background variation, and how we might measure that in the field and how we might understand what we're seeing through that macroscopic lens as well. All right, so some of this work, as we mentioned, started back in 2001. And the very next year, we said, wouldn't it be great if we could go back out and sample a very small subset of those same wetlands and see what happens? What do we think is going to happen? And this was based on some earlier work a few years before that by one of Dr. Cohen's students, Liz Demke, and she was looking at the stability of biological indices. So in this case, something called the Florida Wetland Condition Index. So another researcher, Erica Hernandez, who was going to be a speaker here in a couple of weeks at the Wetlands Seminar, was working on a master's degree and said, what's the community structure stability in the NWCA wetlands? So we had this nice little pool of wetlands. We went back and revisited them the year after, and we found these pie charts, right? I just created these from some of Erica's data, but what we're looking at is on the one side, we have marshes, herbaceous depression wetlands. And if we look at the community structure, so what proportion of the vegetation is a forb or a tree or a grass from year to year, they're very similar, right? So we're not seeing big changes in the actual structural foundation of these sites. Well, it was the same for the floodplains and strands. These are wetlands that have forests, but also connected hydrology. And then also for the forested depressions. The community structure of these is characteristic for that wetland type, but also stable. It doesn't change from year to year. But what was very interesting is that if we looked at a species richness level, about 45% of the species were different on average across the sites in the second year of sampling. So that means if we had a species list of, so many species, half of those were replaced by something in the same growth form or structural class the following sampling period. So that was a really interesting foundation for us. And we thought, how can we look at that with a larger data set like what the NWCA is going to offer us? So one way that we've looked at this is looking specifically at the estuarine wetlands. And we started there because it's a smaller sample size and we wanted to kind of feel for the analyses and how to get going. But also we're interested in coastal dynamics and how those systems might be changing over time. So between the 2011 and 2016 NWCA sampling periods, there were 47 estuarine wetlands that were resampled. It's approximately 10% of that resample total sampling effort. 2/3 of them were marshes and 1/3 of them were considered shrubby. In Florida, you can see on this map here, the ones to the south, those were mangrove. And then as you move up the Carolina coasts in Georgia, those were probably dominated by things like saltbush, Iva, things like that. And then it goes, this data set's cool, right? Because it goes all the way from Maine, around the tip of Florida, down to near Corpus Christi. And then there's 4 points over there on the Pacific Northwest. So it's really widespread. So spatially speaking, we thought, we're going to see really big differences based on where you're located. That's going to be one of the main drivers, right, to how these play out. It did cover 20 coastal states, which was quite a bit. And what you can see is some really fun things, right? So the first thing is when you're in a low marsh, not much changes. And that's good news, right? Because that means probably the hydrology is the same and there's not a lot of disturbance and that kind of thing. So this is an example down by the St. Johns River here in Florida. They went out in 2011. It was 100% Spartina. They went back out in 2016. It was 100% Spartina. So maybe nuanced changes in the coverage, but species-wise, soils-wise, very stable community structure in this system. So then we go over to Louisiana. I'm trying to show you kind of some differences and extremes here in what we're looking at. Again, this is 47 sample sites, all estuarine systems in the NWCA data set that is, I think I forgot to mention, but it's statistically valid, randomly selected sites. So these sites were not handpicked. They weren't picked for certain disturbance reasons. They were very random in that sense, right? So we didn't see a lot of bias, we don't think, in how these sites were selected. But here we are in Louisiana. We have another emergent marsh on the coast. And in 2011, it had 6 species. And when the crew revisited in 2016, it had 29 species. In theory, this is the same exact point in space. Whatever the ability of your GPSs to get you back to that coordinate spot. Same layout for the plots and everything. And so we see just a really dynamic change in this particular system in just 5 years. All right. And then this one, David mentioned, where was I calling in from? So this spot down here in the bottom right of your screen is about 3 miles from where I am now. The aerial photo is Narragansett Bay, so you can kind of see where that is up in Rhode Island. And this site in particular had 8 species in both years, but there was some turnover, right? So one species replaced itself with another one. And so all of that dynamic is very interesting. We use things like species richness and simple diversity indices a lot to characterize communities for their condition. And they're really changing, right? They're very different kind of findings for those. So just at species richness, the marshes had about 8 species on average. The shrub estuarine systems had about twice that and about 16. When we looked at similarities, a number 1 would mean that the species lists were identical from year to year. And so we saw the marshes were about 0.7 and the shrubs were about 0.6. And if you remember the study that we had done on inland wetlands in Florida, that was more like, 0.5, 0.6. So we saw some differences here. I thought it was interesting. Part of it is that those low marshes, if you only have 1 or 2 or 3 species, there's not a lot of variation in that, right? The bottom line was about 2/3 of the species were occurring in the same sampling events. But then it makes you think, if we have biological indicators specific to an individual species and not like a functional type of species, that could be influenced by the turnover in our species lists. We did some cluster analysis on the 2011 data set to kind of understand, thinking that we would see the Pacific Northwest breakout and we would see the Gulf Coast states breakout, but we didn't. There wasn't enough information in the cluster to give us anything that was satisfying as far as understanding wetlands by wetland type or coordinate points, latitude, longitude. And we do think that probably the low marsh was really influential in that, perhaps because some of them were very species poor in general. We took this idea then from those estuarine wetlands and we said, how can we do these just for Florida? Because we do have the resampled data. So looking at all of the Florida resamples, again, it's about 10 to 12 sites from year to year. We found very similar things. These are freshwater and estuarine sites, and we're finding similarity values in the ballpark of 0.6 to 0.8, say. Some of the estuarine sites may be a little higher. I have marked out that the 2021 data are only preliminary. They have not been released at this time. These are not to be taken as the actual values. They're exploratory at this point. The 2021 data will be released all at once from the EPA. So I did want to play around with the data and see where we are and understand what our data are looking like, but still wait on the final publication in order to go forward with that. All right, so thinking about that, we think community structure is stable year over year. Community richness is not stable. Species lists are not stable. How can that help us understand the framework of wetlands, the management of wetlands, and how we might use that in our own human-based endeavors? And that led me to this very interesting question that I've had since we very first started doing site evaluation work with the NWCA. There are a slew of points that come at you. And I think on average, for every 1 site we sample in the field, there are 3 sites that we had to evaluate in the office prior to actually sampling. That's for a whole number of reasons, which are listed here. The site could be dropped because someone didn't want to let you on the property, because it had active crops. Usually in Florida, that has been timber. The area has been bedroad and planted. It could be too deep. And of course, that's highly variable. If we have a wet season, some sites might not be accessible. What we found in 2021 is a few sites we couldn't get to in 2016. We actually could get to in 2021 because the water levels were different. There could be map errors. There are all kinds of reasons. But at the end of the day, we have almost 3 times the data set of wetlands that exist on the landscape, and we don't know anything about them. And I've always wondered, is there bias in that? Is there a reason why people don't let us on their property to sample? Are these the sites that are more impacted by timber production or cropping, or are they in industrial zones and we can't find the landowner? That happens quite a bit as well. So the map here is showing you the green dots are the ones we actually sampled. the squares that are gray are the ones that we dropped. And then there's something called oversample. These are the sites we just didn't need to get to because the EPA only said we had to do 67 sites in this year. And so there's more sites in our data pool that we could also explore as well. So keeping that in mind, what can we do with the data that we're finding in the field and think about the larger data pool for the whole NWCA? And I think there's a role for remote sensing here. And I've played around a little bit with just a pilot study of some of the wetlands in South Florida as an example for this. So some of the ideas comes from the wide availability of data. Landsat 7, their Enhanced Thematic Mapper Plus, has analysis-ready data, and you can get information across the different bands for individual wetland sites. All right, so what does that look like exactly? The graphic here is showing you on the y-axis atmospheric transmission, and across the x-axis we have the wavelength. And so there's peak reflectance for different surfaces at different wavelengths. And that's kind of the graphic that we're trying to see here. It comes from US Geological Survey. And then what I've done is I've basically highlighted the Landsat 7 ETM Plus and tried to pull out the concept idea here. So if you look at the yellow box and look down at those photos, each panel there that's colored is the same exact scene that came from Landsat 7. But what it's showing you is the different type of reflectance you can pull from those images, right? So the first one is the blue, green, red, and then we have infrareds and shortwave infrareds as well. So the idea is, what are we seeing at each point in space in the NWCA database that we can compare back from that remote sensing view down into the actual field data that we've collected? So just to kind of get a feel for some of this information, I've looked at 17 sites in one scene down there in South Florida. You can see the scene highlighted down at the bottom. It includes some of the Everglades areas. And it's a mixture. I just used all the sites that were in it. So there's some freshwater marshes, there's a mangrove and another freshwater shrub as well. And the chart is kind of messy, but I just wanted to give you an idea of what you could see with this information. So each one of the grouped columns is the different type of electromagnetic spectrum that you would be getting. So the blues, the greens, the reds, the near infrareds, and the shortwave infrareds. And each one of the colors is a different site. The idea is we could build an electromagnetic spectrum signature, or an EM signature for each one of the wetland sample points, each one of those AA assessments. We could use our field data to understand them and model it to kind of project out to a lot of the sites that we can't physically get to for reasons, even in years that we don't run the NWCA study as well. All right, so the column over here on my left shows that you're looking at different sites. Each one of those column charts is a different site. They're labeled by their 2011 numbering. And then across the bottom, again, I've stuck with the same color scheme. You see the reflectance values for blues, greens, reds, near infrared, etc. And the idea is, once we get enough of this information, can we take that and ordinate it and try and understand the signatures that we're getting from each of these sites and then overlay that biological data? So in the preliminary work, which is what you're looking at here, this is an NMS ordination. The axes are arbitrary in NMS, but the idea is that we're looking at EM signature space, right? A lot of times we use this for species space, sites per species, but I've used it for its EM signature. And then from there, I've overlaid some vectors from a secondary matrix. In this case, I was thinking, why don't we use something like a biological index? The VMMI stands for the Vegetative Multimetric Index, which is the EPA's method for calculating biological condition for these wetlands. And it has indices like tolerant species, and the FQAI is a floristic index as well. So interesting, you see the grouping, you see the freshwater and the saltwater sites breaking apart, sort of what we expect. Lots more to be done here, right? I think Mark Brown once said something about seminars are, you tell us where you've been, but tell us where you're going. So I hope maybe this is one of those things that we think about where we can move forward with this kind of work. It obviously takes a lot of computing power and time to understand these values and to extract them from the data as well. I did want to mention that one of the areas that I have been following for the NARS is all of the publications that come out from it. And one paper, well, there's 3 papers total that deal with remote sensing that I found for NARS. 2 of them are for lake chlorophyll levels, and one of them is on wetlands. It's a really interesting paper, and if you're interested in this at all, take a look at it. It's in one of the remote sensing journals. They basically took all of the 2011 NWCA data that they could. They excluded a few for reasons of cloud coverage and whatnot on the visuals. And then they took things like richness, and they calculated a standard diversity index as well, and compared it to things like greenness, part of an NDVI matrix, and texture or roughness. The take home for me is that this is a really cool concept, and we can take it and run with it, but they didn't get very far with the plant data. And that's where I think the next step is, right, is trying to understand the intricacies of that community structure, maybe something to do with the proportion of trees and shrubs and vines and how that plays out. Maybe something to do with some of the more complex ways that we can model community structure. If you look at the box and whiskers here, basically across the bottom are all of the wetland types. So herbaceous, woody, and then the first 2 are estuarine and the next 2 are the freshwater. And if you see those, there's no significance here between them. So greenness alone didn't differentiate these wetland types. NIR alone didn't differentiate red variants, etc. So what is it that we can use to tell them apart? Anyway, a very interesting study and something certainly to continue on with. All right, so we've been looking really at that AA scale, the site scale, looking very specific. And then the idea is, what can we do to pull out from that a little bit and to understand landscape differences? There's lots of opportunities here, especially with things like national land use databases and that kind of information, the NDVIs as well. And then we've also done something with the Landscape Development Intensity Index, which is a tool that was developed at the Center for Wetlands with Mark Brown and I think Parker back a while ago, and it's been applied really all over. I just read a paper out of Montana where they were using it. So it's definitely widely dispersed across the US anyway. I think this is an interesting point, and this is one of the reasons why I keep coming back to that question of what about all those data points we're dropping when we can't sample a site, when we can't get access or permission to a site. So for example, we looked at the 2011 data and we found that almost all of the sites had LDIs of 0, and a 0 means natural landscape surrounding it. So in a 100-meter buffer around a wetland site and assessment area, it was all natural wetland or forest or something that didn't get any kind of human land use. There were very few sites, in fact, under 50 that were both an LDI up to 10 and then over 10. So not a lot of these sites are actually occurring where there's human development. And I've always found that an interesting draw within the pool for NWCA, not in any way, to complain about the pool, but just to say, I think that where they lay, we often get interior wetland sites and not always edge. And where we see a lot of change in ecological systems is in that edge effect. So something to consider for sure when we're looking at condition across that landscape. The example here is a site out in New Hampshire. What I'm showing you on the middle picture there is the aerial. The red is the AA, and the yellow is that 100-meter buffer out from the AA. And you can see in this particular tidal creek that there's just a little bit of urban development, some housing that are in there. The bottom panel there is the land use land cover, and you can see that just a little bit of it gets picked up, almost none of it. And some of that has to do with the scale of the grid used in that land use land cover. And some of it just has to do with sometimes the points are offset a little bit from projection to projection for these as well. All right, but in general, I would characterize the NWCA data set as being very heavy on the side of reference sites as a general take on landscape impacts. I did want to let you know, and I won't go into any particular detail about it, but if you are interested in learning more about what NWCA has to offer for some of these other scales of information, here, for example, I've put up the biological and physical alterations These in particular could have been in buffer plots. So we're looking at outside of that assessment area, more of that landscape scale impacts. I think one step for sure could be taking these physical alterations and also perhaps ordinating those with all of our EM signatures and trying to understand the drivers for those different data points as well. And then these data also come from the NWCA 2011, but from Cantula. And I just wanted to point out again, that even though we have this really widespread information on wetland chemistry, especially water, is very challenging to use in part because all of those hashed marks are areas where they didn't have data. So a lot of the sites, for example, by ecoregion, that coastal plain that I have circled in panel A, more than half of the coastal plain sites simply don't have water quality grabbed the day that they're sampled. If we look at the Cowardin classifications for them, almost none of the E2SSs have water taken that day. And if we looked at things like HGM type, so we can see changes in what's available to us and generalizations across the entire US based on this landscape for something like a surface water grab sample could be fairly challenging to take on. And then I wanted to leave you with a couple of graphs just to think about. That first one was pH. Interesting, follows a fairly normal distribution, but things like CEC, cation exchange capacity, and soil lead content, both soil parameters, have very skewed dynamics, right, in their population distributions, and all of that plays into how we have to transform the data in order to use it. I think it's really great to look at this information and try and frame what we expect conditions to be in these systems and when we know we're well outside of that range. And I would say if we look at this phosphorus graph here, we have a fairly normal distribution, and then all of a sudden we don't, right? We have something happening where a lot of sites are actually much higher than other areas. So perhaps those are areas that we can further explore and try and understand that really inside local scale within the AA, but that helps us bridge the systems thinking. So we have our site scale, our local scale, and our landscape scale as well. All right, and so just because I didn't cover it in too much detail, I did want to refer you out to the EPA's data from wetland condition. This is from 2016. For the most part, they have very good findings when it comes to most of their chemical parameters. So for example, heavy metals aren't a concern in most of the wetlands. Nutrients are a concern in less than a quarter. Algae toxins are very low, and microsystems are at less than 1% of wetland areas sampled. So very positive findings as far as wetland condition goes there. And then I definitely wanted to mention that this work is ongoing. If any of you have questions that are in the room or on campus, you can certainly talk to AJ. AJ is working on her dissertation. She was the crew lead for 2021, and she's going to be looking at some of the EPA data over time. Hopefully she'll have the 2021 data to use, so she'll have 3-time series as well. Her focus is really on carbon storage. And so in my mind, that's really sort of this local scale, but she's integrating some of these larger concepts as well, hydrologic positioning in the landscape and vegetation composition as well. All right. And then I did want to leave us some time for questions. So I was going to wrap it up just thinking back at where we've come from with this project. Again, 40 minutes is hardly enough time to cover the NWCA in its entirety, much less the Florida portion of it. We've looked at data from estuarine wetlands across the whole US. We've looked at just some South Florida wetlands. We've looked at North Central Florida wetlands as well. The idea here is that it's really going to take a lot of work to put that detail eliminator in play and to be able to sort through the information that we're getting and to try and use this to really understand the function principles that will help us manage these systems and to help us protect them as well. So our goal for the continuation of this project is really to keep exploring what's happening with the NWCA data, to try and understand the background variability. There's a little bit of thought there on like, what is that range of what's normal in a reference site? What pulsing dynamic keeps systems stable? And then how are we seeing temporal change? Clearly with a changing climate, I don't know if you guys have heard them, but I'm getting weather alerts on my phone right now for flash flood warnings. One of them was going off a minute ago, so I apologize for that. we're having really strange symptoms across the globe. And so we now have a data set through the NWCA and all of the NARS projects that gives us a baseline. We started this over 10 years ago, and now we're going to be able to have this repeatable, statistically valid information in order to come back around to and to continue our understanding from a systems perspective of these wetland systems. All right, I do have a list of references. If you're watching it on the video and you want to grab one of them, I thought I would show them. You can pause it, right, and write them down and grab them as well. And then that was all I had. I wanted to make sure I left a little bit of time. You can reach me at either of the emails that are listed there. If you're on campus, you can get in touch with Dr. Kaplan. You can get in touch with AJ. They know how to reach me as well. So please reach out.
All right, let's give a round of applause to Dr. Reed. Thank you, Kelly. All right, we'll go to questions. I have one from YouTube already, which is a great question. So Charlie Venuto asked, are any of the sample sites in the NWC, are they any human constructed wetlands or like anthropogenically made wetlands? Or are they all natural wetlands?
Yeah, I think that's a great question, Charlie. So I think technically those might have been excluded, but I also think they get included. And so let me qualify that. So there's a particular wetland in Orlando area that I remember us sampling. And it probably used to be a little depressional marsh, and then some construction happened, right? And there's housing all around it. And now it's like a retention basin. And it was still shallow enough to be considered an emergent marsh, but it definitely had been excavated. So by design, constructed wetlands weren't put in it. There's one in the phosphate district, and that area in Florida has just been obviously turned over and reconfigured a lot. And I don't know the site history on that site. It may or may not be a natural wetland or a constructed wetland. We just didn't have enough information. It did get sampled. So I do think that there's a few wetlands that have been impacted by humans in those ways. I think they're pretty few and far between in the larger data set.
Great. Okay, some questions from the room. You guys have been shy in previous weeks, so we're going to just do this uncomfortable silence thing until we get a question from the crowd. But no. We know you have one. We know Dr. Clark has one.
This comforts it.
One of my friends.
Can I answer the question?
All right, AJ, you're going to save the day. Okay, go ahead, AJ.
The question I kind of know the answer to, but the problem I asked, like just not when you're looking at the NWCA and like the spatial distribution of like the number of wetlands across like the country. Why does it look like that? Again, I know the answer, but like it was jarring to me when I first like saw that. It was like, why are they only sampling like 2 wetlands in California, but then we are out there in 60+ wetlands. So like we have, a site every single day. Some people like could go check their site and then come back like a month later and finally decide to sample it. Like, why is the distribution of sites like that across the US?
Yeah, so that's a good question. And it's kind of beyond my role in NWCA, but I will tell you what I know about it. So the site draws come from US Fish and Wildlife Service collaboration with EPA using some of the status and trends reporting information. And that information is not all across the US, right? So it's in certain scenes like the one you saw from the South Florida map. And those scenes occur across the US, but they don't occur everywhere across the US. So one point is that no matter what wetland gets pulled, it has to come from a scene within status and trends or within the data frame they're using. So in 2011, I think everyone recognized that there were a lot of holes. And in 2016, there were a lot of efforts for the next pool to really try and sort of adjust that. So if you noticed in Florida, we had 67 sites to begin with, and we had 48 in the last survey. So they're kind of redistributing them. Some of that is by region, because for their statistical valid design, they need them to be equally distributed across all of the regions and across all of the wetland types. And then some of that has to do, and then for 2021, they added a lot of aerial imagery that states had on hand. So if you had a state and you said, you know what, I think Ohio was one of them. They said, we missed this whole area of the state. And they said, okay, Ohio, or whichever state it was, they said, give us, places that we can pull data from so we can add those into our draws. And so it's an iterative process. The site pools are going to change every year. And I think that's just, it's part of the beast of this massive data pool, you know, trying to figure out where the right data are. Those of you who have worked with NWI coverages, the National Wetland Inventory, you know that they don't always align. There might be a polygon for a wetland that's offset, or that clearly isn't a wetland, or there might be a wetland they didn't get because it was too small for when they originally made those coverages too. So, you know, it's not a perfect fit, but within the context of what's there, it's an iterative process to improve that survey design.
All right, thanks, Kelly. And I think also just the idea that, of course, the places with more wetlands get more sites just because there's a proportional coverage. So like Florida looked like the whole state was covered, and you know, there was like one in, I don't know, New Mexico.
Arizona,
Right?
maybe. Or, yeah. Yeah, I
All
think
right, Mark, go
Kentucky
ahead.
doesn't have a lot of sites either.
So Kelly, great talk and great introduction and overview to a pretty amazing project. And you know, just Florida's again got this heavy infiltration. My question is about, you know, it's probably like 3 or 4 slides from the end there, and there was an identification of a goodness factor for wetlands. For instance, heavy metals, you know, like 85% were good, or you know, only 25% good for nitrogen or something like that. Where is that? How is that determination being made? Because my understanding would be is these very wetlands are the reference wetlands for the most part. They're not, you know, you were talking about looking at, you know, intensity of land use and how that might be impacting. So where are they determining at what point a wetland is good or bad in reference to what? You know, the other reference wetlands, which essentially are these wetlands. So
Yeah, so that's a great question, and I definitely can't address it in 4 minutes, but I will try. So there's a lot of literature out there that explains how these metrics were developed and how they've used different sites. So in the 2011 survey, there were hand-picked reference sites that were not these sites. So there's a whole nother section of the NWCA I didn't even mention that I've only looked at the probability sites, right? So they were looking at the reference sites. So that's one way they were going through these criteria. They also used the data within the probability sites and parsed it out. If you're familiar with some of the ways that the state DEP has used their metrics in the stream condition index, for example, they'll do something where they look at the lowest 25th percentile or the upper 25th percentile and use those as their distinctions within the data sets. And so there's a lot of there's a lot of information on specifically how those are pulled. And I don't know, for example, for heavy metal index, which of those two methods, or if it was a combination of them. But I know in the 2011, there was a huge effort for handpicked reference sites. We contributed a few of those for Florida. Different crews went out and sampled them. So our Florida crew was not responsible for the handpicked reference sites. They had a national crew that went out. They wanted to get a separation of data collection to avoid any kind of site bias. Within their technical manuals, there's definitely this graphic that I'm thinking of that really helps distinguish how they drew those breaks between the reference and then sort of what else you might expect for those condition classes. But they break them out actually between good, fair, and poor. So usually there's multiple watercolor classes. And if you look at some of their sort of shiny sheets that they hand out, they're usually coded like green, yellow, and red, very like intuitive communication strategy so that the goods are green and the reds are poor. And, you know, you can kind of convey that out to the general public.
thank you.
All right, Kelly, just in the moment we have, well, any questions from the group? Anything? Tweak your brains? Okay. Next week, right? Questions. All right, so just as we leave, Kelly, so you taking a look at all these wetlands nationwide and thinking about stressors and disturbance and everything, I guess what would you say the sort of the trend is with wetlands in the US? Are we on good footing? Are there major really like looming issues for wetland quality and wetland integrity? Or do you think we kind of pulled out of the nosedive from the late, from the mid 20th century and we're doing okay? What's your professional assessment on that?
Yeah, well, I think that's a loaded question, David. It's like,
It's
all
being
right, so
recorded.
I know, right? For everyone to hear. Yeah, so part of the rating for NWCA comes back, and I forget the number exactly, but it's somewhere in the ballpark of 45 to 48% of wetlands are considered good, biologically speaking. So that alone tells me that we have a ways to go. the EPA does offer some data dashboards. I couldn't, the wetlands one is a little bit behind because it was the last of the 5 surveys to be reiterated. So it will eventually connect so that you can do change. So on their data platforms, you can actually see an improvement or a decrease in condition across all the parameters. If you went today to NRSA, the National Rivers and Streams, you'd be able to see that very answer, right? It'll say good direction, bad direction, that kind of thing. The major impacts to wetlands that they're finding is surface hardening, changes in water, vegetation removal. So a lot of physical alterations are really changing the landscape of the wetlands. We don't have the 2021 data in hand, so we really only are looking backwards from 2011 and 2016. I don't know what the 2021 data is going to look like, you know, as far as like, are we seeing similar trends for, I don't personally think that 45 or 48% of wetlands in good condition is a particularly strong testament to our wetland condition. That being said, I don't have data from 1990 to see, you know, what that's changed either. So this is a great baseline. You know, we're starting in 2011. We've manipulated a little bit the methods and the survey design, just the site selection, and it's improving every iteration. And I think we have to wait and see what the data tells. It's a safe answer.
All right. Yeah, thanks, Kelly. All right, let's give Dr. Reese another round of applause. Before we go, just we'll see you all next week. Our speaker next week is going to be live and in person. Here is coming up from Tampa, Dr. Mauricio Arias, and he's going to talk about a new generation of computer models to manage Lake Okeechobee's harmful algal bloom. So very close to home, very relevant, and he'll be here. So everyone on Zoom, everyone here should be here in the room. All right, thank you, Kelly. Appreciate you.
All right.
See you later.